everyone, welcome back to my channel, and today we are going to learn something interesting in enzymes. That is actually the you can say the helper of enzymes, or you can say the how enzyme is going to be used. So we have, like, you can say, two helping agents for enzymes. One is known as coenzyme, other is known as prosthetic group. So when we talk about coenzymes are basically loosely attached to the enzymes. And what do? They bind to the active site. Sorry, they bind the substrate with the active site, and they are loosely attached. They are not permanent part of the enzyme, but they help actually the substrate, and they show them a way, or they can just help them in some induce some change in the structure, in the conformation, so that they can easily fit into the active site. While the other one is the prosthetic group. For example, there are certain ions, or you can say minerals, that are very essential for the enzymes to work. For example, potassium or magnesium. So these one are permanently bound to an enzyme, and they contribute to the final 3D structure, that is the tertiary structure. Then we have free and immobilized enzymes in the use. And how are we going to use them? These are extensively used. I'll give you only one example. So let's just go into the definition. Free means free, mean it is not attached to something, and they are added directly to the solution. The substrate can be added to enzyme solution, or the enzyme can be added to substrate solution. Simple. While immobilized mean they are like attached to something. They are bind to inert, stationary, insoluble material, for example, alginate, and the substrate part pass over these immobilized enzymes, and the product is collected. Now, how is it used? You might have heard of lactose intolerance. It's quite common nowadays. And we have lactose free products. Now, how those products are made? So, the answer is the immobilized enzymes. Now, this is the general process. What is going to happen? This is alginate in on which your enzyme is actually embedded. So when you pass, for example, you pass milk over here, which contains lactose, let's say milk lactose, and someone is lactose intolerant, that lactose must be broken down. So when it is passed and it moved through these immobilized enzymes, the product is collected over here, which would be galactose and glucose. So we have galactose and we have glucose. Now this one means the lactose is broken down. This one is not needed to be removed because they are already present in some other form. So this is the main advantage of immobilized one. So product is uncontaminated by enzymes. Now this product doesn't contain any enzyme because enzyme is present on beads. You can easily say this. So no filtration is needed, first thing. Otherwise filtration is really hard process for enzymes. Enzymes can be reused. It's cost effective because you need to use this column again and again. Great tolerance to pH and temperature changes. It's more stable, and it is used in the food industry to produce lactose free milk. Milk is passed over immobilized lactase. The lactose hydrolyzed into glucose and galactose. Simple. If you want to know what is hydrolysis, you need to go to the video of carbohydrates, that how the bonds are going to break of lactose molecules, that is through hydrolysis. Go to that video and you can learn from there. See you in the next video.